Our protocol provides an effective and repeatable approach to evaluate and quantify phagocytic events in hemocytes. The main advantage of this technique is that we can quantify phagocytic events within individual hemocytes, which allows us to detect variations in phagocytic processes among genotypes and individual flies. Performing dissections can be challenging, so when performing this technique for the first time, practice dissections as much as possible before carrying out the full procedure.
Begin by preparing glass needles for injection. Fill a 1 mm syringe with mineral oil and attach the 30-gauge hypodermic needle provided with the nanoinjector. Insert the 30-gauge needle into the blunt end of a pulled capillary needle and fill it with mineral oil. Slowly remove the 30-gauge needle, making sure that no air bubbles are formed. Remove the collet and place the sealed O-ring with the indentation facing up. Place the larger O-ring onto the metal plunger. Then reattach the collet without tightening. Insert the metal plunger into the blunt end of the oil-filled glass needle and gently push it down, inserting it into the larger O-ring. Then tighten the collet until secure. After filling the needle with fluoro or pH-sensitive particles, inject the flies in the sternopleural plate of the thorax. The injection is successful if the green dye is going into the fly. Place the injected flies into a new food vial, noting the time that the first and last fly were injected. Lay the vial on its side until all flies have recovered to prevent them from getting stuck.